Hello everyone, welcome you all in the EPG Pathshala program in the subject Botany. I will talk on salicylic acid. Salicylic acid is a phenolic phytohormone. It was first isolated from the extract of bark of white willow Salix alba. This plant hormone plays a crucial role in plants' endogenous signaling involved in mounting the local as well as systemic acquired resistance against pathogen. In this module, we will study structure and biosynthesis of salicylic acid, its metabolism, catabolism, transport, physiological effects and mechanism of gene regulation. Salicylic acid is a phenolic phytohormone which was first isolated from the extract of bark of white willow Salix alba. Chemically, it is hydroxybenzoic acid. Salicylic acid plays a crucial role in plants' endogenous signaling involved in mounting the local as well as systemic acquired resistance against pathogen. Salicylic acid is chemically a hydroxybenzoic acid. Salicylic acid is synthesized in plant via two different pathways, phenylalanine ammonia lyase, that is PAL pathway, and isochorismate pathway, that is IC pathway. Chorismic acid, the end product of shikimic acid pathway, is the main precursor of both PAL and IC pathways. First, I will talk about PAL pathway. Phenylalanine ammonia lyase or PAL is the first enzyme of phenylpropanoid pathway. PAL converts phenylalanine to trans-cinnamic acid and ammonia via non-oxidative deamination reaction. Trans-cinnamic acid is converted to orthochimeric acid, benzoic acid or cinnamol CoA, the three possible intermediates of salicylic acid depending on the plant species. Decarboxylation of orthochimeric acid produces salicylic acid. Ba could be formed by three different routes. 1. By non-oxidative OA from trans-cinnamic acid. 2. By beta oxidative OA from cinnamol CoA and by non-oxidative OA from cinnamol CoA. Further, Ba is converted to salicylic acid by an inducible Ba2 hydrolase. IC pathway. In the bacterial species, chorismate is isomerized to IC by a unifunctional enzyme, isochorismate synthase or ICS. IC is further converted to salicylic acid and pyruvate by the enzyme isochorismate pyruvate lyase. Alternatively, chorismate is directly converted to SA by a bifunctional enzyme salicylic acid synthase via an isochorismate intermediate. In plants, chorismate is synthesized in the chloroplast. In 2001, two putative ICS genes were identified from Arabidopsis genome. Biochemical and molecular analysis of ICS1 proved the existence of IC pathway in planta. Salicylic acid, the produced upon pathogen infection or UV exposure, is mainly via IC pathway. Now, we will see how salicylic acid is metabolized, transported and stored in the plant. Salicylic acid is glucosylated, methylated, sulfonated or conjugated with different amino acids. Salicylic acid becomes inactive after most of these modifications. However, the inactivation helps in the fine adjustment in the accumulation, function, homeostasis and mobility of salicylic acid. The first modification is glucosylation. Salicylic acid is glucosylated by the enzyme UDP glucosyl transferase. In Arabidopsis, two UGTs, that is UGT74F1 and UGT74F2 catalyze the conjugation of salicylic acid and finally form SA2O beta glucosidase. Conjugation occurs at the hydroxyl group. However, UGT74F2 can also catalyze the conjugation of SA at the carboxyl group to form a salicylate glucose ester. In Arabidopsis, tobacco and soybean, SA is glucosylated to the non-toxic SAG in the cytosol and then it is transported to the vacuole for storage. Glucosylation inactivates salicylic acid, but SAG can be hydrolyzed to liberate salicylic acid for inducing defense responses upon the pathogen attack. SA is also methylated to its methyl ester, methyl salicylate, by the enzyme BA or SA carboxymethyl transferase. Generally, expression of BSMT1 is higher in the flowers compared to the leaves. It is responsible for floral scent, which attracts pollinators. Upon biotic or abiotic stress, the expression of BSMT1 is induced in leaves, resulting in increased methyl salicylate accumulation. Methyl salicylate is not biologically active. Newly formed methyl salicylate is transported to the systemic leaves via phloem and is converted back to salicylic acid by methyl esterase activity of SA binding protein 2. Upon pathogen infection, the glucosylated form of SA is also methylated to form MESAG by the enzyme salicylic acid glucosyl transferase. The function of MESAG is not yet known. However, it is thought to be a non-volatile storage form of methyl salicylate. Unlike jasmonic acid, very less is known about salicylic acid amino acid conjugates. In Arabidopsis, Phaseolus and in Vitis species, SAASP has been identified as a stable and major salicylic acid amino acid conjugate. In Arabidopsis, it has been shown that GH3.5 enzyme is responsible for salicylic acid amino acid conjugation. However, exogenous treatment with SAASP is unable to induce any pathogen related gene expression, suggesting that SAASP is an inactive form. Sulfonation can both activate or deactivate the hormones. It has been shown that the sulfotransferases catalyze the transfer of sulfonyl group to the 2-hydroxy position of salicylic acid and salicylic acid is sulfonated. SA is also catabolized to form dihydroxy benzoates. Recently, it has been found that salicylic acid is catabolized to 2,3-DHBA by the enzyme SA3 hydrolase during leaf senescence. This enzyme converts SA to both 2,3 and 2,5-DHBA in an in vitro reaction. However, in vivo it converts SA only to 2,3-DHBA. The formation of DHBA is induced by SA. Therefore, the formation of DHBA acts as a negative feedback which prevents in the planta over accumulation of salicylic acid. Apart from being a key signal in the regulation of thermogenesis and disease resistance, salicylic acid directly and indirectly influences various vital processes such as seed germination, seedling establishment, cell growth, respiration, stomatal closure, senescence associated gene expression, thermotolerance, nodulation in legumes, fruit yield and responses to abiotic stresses. Thermogenesis. The reproductive structures of cycads and flowers of some angiosperm produce heat. This phenomenon is called as thermogenesis. Production of heat facilitates pollination by spreading the chemicals that attract pollinators to the plant. An increase in SA level in the plant or exogenous application of SA induces thermogenesis. Salicylic acid increases the activity of the alternative respiratory pathway in the mitochondria by inducing the expression of alternative oxidase, the terminal electron acceptor of this pathway. The electron flow through this alternative pathway generates ATP at only one site, and unused potential energy is released as heat. Disease resistance. After the perception of an invading pathogen attack, plant mounts effector triggered immunity, that is ETI, by establishing a direct or indirect interaction between the product of host resistance gene and the corresponding pathogen encoded effector protein. ETI results in the hypersensitive response where plant develops a necrotic lesion at the site of pathogen entry. ETI is also associated with the accumulation of reactive oxygen species and induction of various defense related genes, including the genes encoded for pathogenesis related proteins. Plant also mount the pathogen associated molecular pattern, that is PAMP triggered immunity, a surface receptor based pathogen recognition system. PTI creates a low level resistance to the virulent pathogens. Salicylic acid is an important signal for the activation of both ETI and PTI. Now, we will talk about SA signaling and mechanism of gene regulation. For SAR development, signals from the infected leaf need to be transmitted in the uninfected parts of plant. Signal transmission takes place via phloem. SA and MESA have been found to be the major and long distance signals respectively. SA signaling network is very complex and divided into upstream and downstream signals. First, I will talk about upstream events in SA signaling. Two major subsets of resistant related genes, that is R genes, namely tall interleukin 1 receptor nucleotide binding site leucine rich repeat and coiled nucleotide binding site leucine rich repeat. These are abbreviated as TIR-NBS LRR and CC-NBS LRR respectively, have been identified as upstream players to SA signaling. Lipase-like protein enhanced disease susceptibility 1, that is EDS1, and its interacting partner phytoalexin deficient 4, that is PAD4, act upstream of SA in the ETI initiated by the TIR-NBS LRR subset of R genes. EDS1 and PAD4 positively regulate SA signaling. Translocation of EDS1 complexes between nucleus and cytoplasm is important for the effective disease resistant signaling. The second subset of R genes, CC-NBS LRR, is regulated by non-specific disease resistant 1, that is NDR1. NDR1 is a glycophosphatidylenoacetal inositol anchored plasmamembrane protein which regulates the R protein signaling via RPM interaction protein R, that is RIN4. Calcium and calmodulin also play important roles in the regulation of salicylic acid accumulation and signaling. In Arabidopsis, binding of calcium or calmodulin to the transcription factor SR1 impairs EDS1 expression and suppresses SA accumulation and defense. Now, I will talk about downstream signaling. Downstream SA signaling involves salicylic acid protein and protein-protein interaction and induction of genes. First, I will talk on SA protein interaction. In tobacco, 4 SA binding proteins have been identified. Among these, SA binding protein SABP2 shows the highest affinity towards SA. it was found that SABP2 is required for both local resistance and SAR. Binding of SA to SABP2 active site inhibits the function of methyl salicylate esterase and enhances MESA accumulation in the infected tissue. Later, this accumulated MESA is transported to the non-infected plant parts. Another SA binding protein is catalase, which is responsible for the degradation of H2O2. However, an increase in SA level or an application of biologically active SA analog inhibits the H2O2 degrading activity of catalase. In addition, SA also impairs the activity of another H2O2 scavenger, cytosolic ascorbate peroxidase. Tobacco chloroplastic carbonic anhydrase CA is another SA binding protein that plays a role in plant immunity. Nitrosylation inhibits CA's SA binding ability and enzymatic activity. Therefore, nitrosylation of CA is thought to be a negative feedback regulation that mediates defense responses. Next is protein-protein interaction. Non-expressor of pathogenesis related genes, that is NPR1, is the main regulator of signaling downstream of SA. It is an ankyrin repeat containing protein with a BTB or POZ domain. In the cytosol, NPR1 oligomers are held together by the disulfide bridges. Salicylic acid induced changes in the cellular redox state reduce 2-cysteine residue by thioredoxin H5 or H3 and monomerizes NPR1. Finally, these monomers are translocated from cytosol to nucleus and activate the transcription of defense genes. In feedback, NPR1 negatively regulates the accumulation of SA by repressing the expression of ICS1. The negative feedback regulation is important for SA homeostasis. Now, let us see how the genes are induced. Genes acting downstream of SA induction are classified in two groups. Genes which are induced within 30 minutes of SA elicitation are called immediate early, and ones induced after 30 minutes are called late genes. Induction of immediate early genes are NPR1 dependent. Their NPR1 dependence increases with time. PR1 is the most studied SA inducible late gene. It is regulated by the NPR1 interacting TGA factors. NPR1 and SA promote the binding of TGA2 to the promoter of PR1. Interaction between NPR1 and TGA2 leads to the formation of enhantiosome, in which C terminal of NPR1 acts as transcriptional transactivator. The Werke family of transcription factors act downstream of SA signaling. Werke 18, 28, 53, 54 and 70 act as positive regulators of SA mediated resistance. Among them, Werke 28 binds to the W box core motif present on ICS1 promoter and works as a positive regulator of induced ICS1 expression. Other transcription factors, namely ethylene response factors and R2-R3 MYB factors, also affect SA signaling. Now, crosstalk between salicylic acid pathway and other signaling pathways. SA pathway interacts with several other signaling pathways. SA antagonizes jasmonic acid, auxin and abscisic acid mediated signaling. In general, SA signaling suppresses JA dependent genes and vice versa. SA-JA crosstalk is partially regulated by the redox state of the cell. In Arabidopsis, SA induces the expression of glutaredoxin 480 in NPR1 dependent manner. Later, GRX 480 interacts with TGA factors and also represses the expression of JA induced defense gene plant defensin 1.2, that is PDF 1.2. Werke transcription factors also influences SA-JA crosstalk. Overexpression of Werke 70 enhances the SA responsive PR genes and at the same time suppresses PDF 1.2. In Arabidopsis, mitogen activated protein kinase 4 negatively regulates SA signaling and positively regulates JA signaling. SA also antagonizes auxin signaling. Conjugation with amino acids inactivates auxins. The enzyme GH 3.5 is responsible for the conjugation of both SA and auxin with amino acids and attenuated auxins activity. Abscisic acid indirectly affects SA signaling via its effect on jasmonic acid signaling or vice versa. Now, to summarize, salicylic acid is an important plant hormone. Chemically, it is hydroxybenzoic acid and biosynthesized by PAL or IC pathway. Salicylic acid can be modified by glucosylation, methylation, amino acid conjugation and sulfonation. SA can also convert it to dihydroxyl benzoate. Salicylic acid plays a significant role in local as well as systemic acquired resistance against pathogen. Thank you.